Okay. There was somebody saying something. Who was it? Yes, Peter. Someone was saying something. Can we start from there only? Who was it? Some query was there.
Yes, ma'am. Ma'am, I was asking that in these type of proteins, there is no hydrogen bond in
Beta,
tertiary.
beta, all, see, I'll be coming to proteins. Now you just tell me one thing. You can expect the chains to be formed without hydrogen bond. You think closer आएंगे जब amino acids तो bonds होंगे नहीं होंगे बेटा उनके बीच में?
Yes, ma'am.
100% होंगे. We do have bonds. Very good question. I'm just coming to that. मैंने लिख रखा है. Next, after I do the structure, I've taken a separate paragraph to explain the bonds to you. If you can see on the screen over here, primary, secondary, tertiary, and quaternary. Okay, we're just coming to this, but very good. Hydrogen bonds are there. So we're coming to the next structure. I was talking about these enzymes. We know about protein digestive enzymes. Have you heard about protein digesting enzymes? कभी तो word पढ़ा होगा protein digesting enzymes.
Yes, ma'am.
Yes. Why only proteins? Why don't they digest other things? Carbohydrates को क्यों नहीं digest कर लेते हैं? There are some enzymes which we call them as protein digesting enzymes. Why don't they digest other things like carbohydrates or something else, lipids? Why only proteins they digest? The reason is they are specific proteins. Got it? They will only be able to bind with whom? A specific substrate, and then they are able to digest it. These kind of proteins, we call them as what? We call them as protein digesting enzymes. Okay, because they have specific. Example of tertiary proteins are enzymes. What is the example of tertiary proteins?
Enzymes.
That is why we say that enzymes are very specific in their action. They just do not act on any substrate. They are very specific because they have to combine with both the groups. We're coming to the fourth one, that is your quaternary proteins. If you can see, we've got 4 chains over here. This is 1 chain, 2 chain, 3 chain, 4 chain. 2 verticals, 2 horizontals. So we have got the next structure, that is your quaternary proteins. Okay. They are also specifically functional. The example is the hemoglobin. I just told you when I started in the beginning, the globin part of hemoglobin. How many alpha chains, how many beta chains? When I started the topic today, I told you. How many were
2
there,
alpha
beta?
and 2
Two
beta.
each.
So you can see total number of 4 chains over here are 4. So hemoglobin, that is the globin part of hemoglobin, is an example of which protein? Primary, secondary, tertiary, or quaternary?
Quaternary.
Quaternary.
Because it has got how many chains?
4 chains.
4.
Chains.
2 alpha and 2 beta. This is also a specific kind of a protein, because if you remember, आपने पढ़ा होगा carboxy hemoglobin, oxy hemoglobin, पढ़ा है ना?
Yes, ma'am.
कभी
Yes,
ये पढ़ा? Ammonia hemoglobin. Did you read about this word anywhere? Sulfur hemoglobin?
No, ma'am.
No, na?
No, ma'am.
That means hemoglobin. That means your hemoglobin is combining with oxygen, carbon dioxide, and of course at times carbon monoxide also. Carboxy monohemoglobin, carboxy hemoglobin, oxyhemoglobin. So basically it is combining with only 3 gases: O2, CO2 or CO. This shows that it is specific in its action, hemoglobin. हमने कहीं नहीं पढ़ा, हमने कहीं नहीं सुना sulfur hemoglobin. We have never seen hemoglobin combining with sulfur or phosphorus or sodium or chlorine, because it is specific in its action. And it is specific because it is which kind of a protein? It's a quaternary protein. Okay. So the example for this quaternary proteins is which one? I just told you it is hemoglobin. Now let's come on to the same thing. Bonds. If I talk about the primary structure, we had only one bond. Which was it?
Peptide.
Peptide bond.
Let's move on to the secondary structure. We had 2 kinds of bonds. What are they?
Peptide.
Peptide and hydrogen.
In tertiary, we will have peptide bonds, we will have hydrogen bonds. We may also have ionic bonds. Okay, at times we may have which kind of bonds? Ionic bonds and disulfide bonds. Disulfide bonds kab honge beta? If you have an amino acid which contains sulfur, both the amino acids contain sulfur, then the bonds which will be formed between them एक और bond बन जाएंगे extra. Apart from peptide and hydrogen, वो कौन से bonds होंगे? Disulfide bonds. But disulfide bonds सिर्फ तब बनेंगे when two sulfur containing amino acids will come closer to each other. Two chains of amino acids or two amino acids which have sulfur, both of them have sulfur. तो sulfur sulfur के बीच में एक और bond बन जाएगा. This is disulfide bond. But बेटा ये third bond होगा. Peptide bond तो essential है. Amino to amino जुड़ेगा तभी protein बनेगा. And helix structure में जाएंगे तो hydrogen bond तो है ही है. And this possibility is also there that there is a disulfide bond. But remember, this is a possibility. Next is quaternary structure, peptide bond, hydrogen bond, and ionic disulfide bonds. Again, the same case. If your amino acids have sulfur, then both the amino acids which are bonding have sulfur, then additional एक bond बन जाता है. That is which bond? It is your disulfide bond. Which kind of a bond is this? Disulfide bond. The last classification that I'm taking up is the shape, based on the shape of the protein. This is the last part, beta. On the basis of shape, we have got two kinds of proteins, fibrous and globular proteins. Fibrous proteins are the spiral polypeptide chain. Spiral, you remember? हमने बनाया था अभी एक figure में. आप पहचानो?
Yes,
This,
ma'am.
yes, these are spiral.
Yes, ma'am.
So when we're going to shapes, we've got how many categories of proteins based on shapes?
Two.
Two.
And globular.
Yes,
Two.
I'll tell you about the fibrous one. They're spiral polypeptide chains wound around each other to resemble fibers. They look like what? They look like fibers after they wound around each other. Generally, they are insoluble in water. Fibrous proteins are generally insoluble in water, but they're soluble in concentrated acids and alkalis. Okay? They are soluble in concentrated acids and alkalis. Example is keratin of your hair. Hair keratin is insoluble in water, but it is soluble in acids and alkalis. So this was the first shape of your proteins. Second is your globular. The word globule tells you that it is round in shape. ये उल्टा है बेटा. They are generally soluble in water. They are also soluble in dilute acids, alkalis, and salts. Additional thing in globular is that they are soluble in water also. Example is your egg albumin. What is the example of globular protein?
Egg albumin. Egg albumin.
What example did we do of fibrous protein?
Keratin of hair.
Keratin of hair.
So functions we know, they help in transportation of nutrients. They act as plant protein transporters. They help in the movement of muscles. You remember myosin and actin. If you have read about these proteins, myosin and actin, they help in the movement of muscles. Proteins help in the growth and repair of the bodies, and they also act like hormones or enzymes. So very quickly, what we have done today, which biomolecule have we studied today?
Proteins.
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 Proteins, why the word protein comes from the word?
Proto.
Proto, which means
What proto means?
prime importance.
Yes. All the study was done by Mulder, but the term was given by?
Virgilis.
We had another name of proteins, that is polypeptides. It could have one polypeptide, it could have many polypeptides. So the first classification we did was based on the number of polypeptides. How many groups we had? Two. One was monomeric. The other was
Multimeric.
multimeric. So if there's just one polypeptide, it is monomeric. What example we did?
Lysozyme.
Yes, lysozyme. It is found in which secretions?
Saliva.
And tears.
Very good, बहुत बढ़िया बेटा. And if it has got many proteins, then we call it as which kind of a protein?
Multimeric.
We did the example was the globin part of hemoglobin. How many chains are there?
Four chains. Two alpha and two beta.
Alpha
And
beta.
two beta. So you put it in what category? You put it in multimeric or monomeric?
Multimeric.
One more example of multimeric protein we did was
Insulin.
Insulin. The inactive form of insulin is called?
Proinsulin.
Proinsulin has how many chains?
Three chains.
And the active insulin has?
Two chains.
But it is, both of them are more than one. So we will put them in which category of proteins? Multimeric or monomeric?
Multimeric.
Second classification we did was based on the content of the proteins. First class proteins and
Second class proteins.
Second class proteins.
First class proteins are also known as complete proteins. Why are they called complete proteins?
Because they contain all kind of
contains all
amino acids, whether
Amino
essential
acids.
or non-essential.
essential or
Very good, very
non-essential.
Very good. They give the body, they contain essential, non-essential, all amino acids. So the complete nutrient content is satisfied. What was the example we did for complete proteins or first class proteins?
Animal protein.
Animal proteins.
Yes, we had just discussed about animal protein, that is meat and egg. The second part was your second class proteins or your incomplete proteins. Why do we call them as incomplete proteins?
Because
Because
either
They
has
lack some of the
only
amino acids,
essential
essential or
or
non-essential.
non-essential.
They lack any of the amino acids. And what was the example of the second class proteins or incomplete proteins?
plant protein.
Yes, beta, the plant proteins. So the plant proteins are incomplete proteins, or you call them as second class proteins. Then we did classification on the basis of structure. This is the third classification. If it's a linear chain, then it is which structure?
Primary structure.
Primary structure.
Primary structure, the proteins are non-functional and there is only one bond present between the amino acids. What is that bond?
Peptide bond.
Peptide bond.
Peptide bond. So I said generally ये होता है कि primary structure is inactive, non-functional, but there is one exception. What exception I give you?
Insulin.
Insulin.
Very good. So let's talk about the secondary structure. In secondary structure, we had two bonds. Peptide bond के अलावा एक और bond बना था, and that was
Hydrogen bond.
Hydrogen bond.
If hydrogen bond is formed between the same chain, we get this structure. What is the structure called as?
Alpha
Alpha helix
helix.
structure.
Alpha helix. The example was
Keratin.
Keratin.
And sometimes you may have these kind of bonds where the hydrogen bond is formed between two different chains. Then such a structure is known as
Beta.
Plated.
beta plated structure. Example was what?
Silk fiber.
Silk fiber.
Fiber.
So with your tertiary proteins, you have three chains together. And the most important thing about tertiary and quaternary are that they're specific. They are very specific in their action. We call them specifically functional. That is why this protein, tertiary protein, is also called as which kind of a protein?
Specific.
Specific protein. And the example are enzymes. What is the best example of tertiary proteins?
Enzymes.
Because they're very specific. They're very specific in their function. Quaternary proteins pe chalte hain beta. How many chains were there?
4 chains.
4. We have already discussed the example of quaternary proteins. It was globin of hemoglobin, which had 2 alpha and 2 beta chains. Very quickly, we did bonds. Primary structure, we had only one bond, peptide. Baki tertiary, quaternary, subme, secondary, we are going to have peptide plus hydrogen. However, tertiary and quaternary, we may have disulfide bonds also. ये तब होगा when amino acids containing what are joining together?
Sulfur.
Sulfur.
Last classification was based on the shape, fibrous and globular. If you have a spiral shape wound around each other, then this is which shape?
Fibrous.
Fibrous.
Are they soluble in water?
No.
No.
ma'am.
The example is?
Keratin of hair.
Keratin of hair.
When we said globular, what is the shape of globular?
Round in shape.
Rounded shape.
They are soluble in water, salts, acids, alkali, soluble. Okay, what was the example?
Egg albumin.
Albumin of
Very
egg.
good. I hope the concept is clear. The basic structure of proteins and the 4 types of classification that we have done on the proteins. Number of polypeptides we did the shape also, we did the structures also, and we did the content classification. Keep revising what we have done, children. Then tomorrow we'll take up another kind of biomolecule. Okay? Keep revising. God bless you all.
Thank
Thank you, ma'am.
Thank you, ma'am.
Thank you,
Bye,
ma'am.
beta. Bye, bye, bye. God bless.